Here, everybody. Welcome. Welcome to Lincoln University. Welcome to the Center of Excellence for Transformative Agribusiness. My name is Jim de Koning, and together with Ellen Renwick, we both are the directors of the Center of Excellence. Center of Excellence is actually a real unique is a unique center because we are multi-faculty. So all the associates that are within the center are can be from all faculties. So we try to look at things multidisciplinary, transformative agribusiness. So we basically assume that agribusiness, what that we're going to do tomorrow, will be different than we're doing it today. We're focusing on four areas. Alternative proteins, alternative energies, circular economy, and sustainable food supply chains. Today we have a very special guest, Associate Professor Dr. Sharma, come here from India. He will be with us for about 2 weeks, and so we are really stoked to have him. He's doing research in the area of medicinal plants and polyphenols and other some probiotics, I believe. So he will take us on a journey there. Before I give him the floor, I just wanted to do some housekeeping. We do not expect any exercises today. So if the alarm goes, it is the real deal. The door that you came in through is one of the exits, and in the back of the room there is an exit as well. We will gather in front of the grass between the lodge and the Ross building, but there are plenty of Lincoln University staff in the room to actually escort you out there. Toilets, this door, turn to your left, and just follow the signs for toilets. But without any further ado, Ms. Sharma, floor is yours.
All right, ladies and gentlemen, good afternoon. I think I'm audible. Yes. Thank you. Thank you for being here. And I'll be talking a little bit about what I have been doing and how it could be useful or pertinent to the New Zealand ecosystem as well. So my talk is related to amalgamation of polyphenols and probiotics as functional foods. So I'll be discussing a brief overview of how we are combining these two elements together and what research are we doing in that respect. We know functional foods provides some health benefits beyond basic nutrition. We are all aware of that. We regularly consume them. Amongst those two elements, probiotics and polyphenols make a large chunk of it. So whatever we consume daily, in one form or the another, in terms of functional food, we will have these two components. So one basic theme would be how they interact with each other and how can we create the so-called good or superfoods combining these two elements. So they are the most bioactive ingredients as far as the functional foods are concerned. These foods can be natural or fortified. So we are trying to make fortified foods. Naturally, they exist, pomegranates, berries, you name it, all those sort of stuff will be classified as natural functional foods. So if you go to the individual ingredients, probiotics, we are well aware, live microbes. We consume them daily in one form or the another. They have lots of beneficial effects, particularly related to our nutrition, metabolism, the absorption of our nutritional elements, maturation of the immune system, and my favorite subject, that is aging. So we'll come to that in a moment. And the foods that we have talked, we are aware of this along with the commercially available things. So they provide a lot of probiotics, live microbes. The issues related with them are strain specificity and individual variation. So that means, although there might not be one good bacteria fits all, right? So I'm not going to detail of all this. We are aware of these. And then polyphenols, diversity is the keyword associated with them, as is with the probiotics. They're very diverse. The thing is they possess strong pharmacological effects. They have the ability to modulate cellular systems, cellular cycles, cellular effects, and have real world effects in terms of diseases or in terms of cell functions. Their daily consumption is associated with a decrease in all-cause mortality rates. One thing I'd like to add here is, for example, in Japan, people live long enough, very long. So it may not be a causation, but at least a causality that they consume 4 times more polyphenols than the rest of the world on a global average. So daily consumption of these in raw forms is very much appreciated. Their issues are bioavailability. So that means that in cells, they might be very potent, but when you consume them, you don't get enough so that they may have a real world effect. So we need to figure out these so that the dosage related concerns are taken care of. Again, lot of things they do, lot of things. We'll discuss few of those as we move along. So I'll be discussing more on healthy aging. My core area of research relies on understanding what is aging and what we can do about it. So the global elderly population is on the rise, including here in New Zealand. So we need to figure out what kind of specific nutrition can we have to promote the concept of healthy. So before I move further, what actually is a fantastic subject, both philosophically as well as scientifically. So what it is, where does it occur, how does it occur, why does it occur? There are lots of questions. There are lots of theories, unfortunately not yet a universal answer, but we have something called a breakthrough research done relatively recently, and they identified what is called a senescent cell. Now, this is a naturally formed cell in every organism, and every organism that counters aging has been found to have less of these cells. For example, the anomaly in aging world is something called a naked mole rat. It lives much beyond than it is supposed to be based upon its body weight. And it was recently found that these guys take care of these cells so that they can live longer. So we might have what is the golden recipe, or at least what a scientist believing in this might like you to believe as well. But still, there is not a universal theory of it. But you can, as you can see here, when we are young, we have lesser of these cells. When you get old, we have more of these cells. So you can see if you have the root cause, you can target these cells. You can say probably, okay, I'll take care of these cells, and then I will at least be able to enhance what is called the health span, right? So you might want to live longer, but you don't want to live, with disease. That's not a life. So health span is also associated with this. So you can see these cells develop naturally is another important thing to understand. These cells develop naturally, and they may have far more role in aging that we may initially have thought of. So targeting these cells through natural means, through diets, is what I'm interested in. So these can happen because of so many various reasons, and as they accumulate, they can create lots of problems. One more thing, zero science hypothesis. Now, until now, we have funds for we study cancer in a different way. We study diabetes in a different way. We study arthritis in a different way. Neurological disorders, all are separate entities, so to speak. But this hypothesis, it's a very elegant hypothesis because it states that all diseases at the root cause lies the presence of senescent cells. So if you think about it, these diseases more or less are associated with advanced age. So if you remove these cells, all those diseases are taken care of. So there is a great accumulating evidence in that respect. So that makes this field very interesting, and that also makes senescent cells a potential target for natural therapies through types. So we developed a protocol for senescence to understand this, how to make a cell senescent. The moment it divides, it is not senescent. So we had to develop a protocol. So we developed this method for HTS. We can have high throughput screening of different functional food elements. So you can actually create a cell senescent in 7 days by giving it stress. So this is an accelerated model of aging, right? So how does it look like once you're done with the technicalities? I just wanted to show you this. So this is how a cell will look like. You can see the size here. This here is the normal cell. I'm talking about fibroblasts. This is the adipose tissue cells. So you can see these cells are pretty small. But when you see here, look at this cell. Starts from here and expands all the way down. This is how a senescent cell looks like. They tend to be very large and flattened in morphology. So this is a potential target. So the mechanisms to understand this is how you want to target through therapy, which I'll come in a moment. So this is how we can develop a method or a model to study them. There are many ways to characterize them. One way is this. You can see very clearly. This is a control cell. This is senescent cell. You can clearly see some markers green. I think these are visible. This is what is called a senescence-associated beta-galactosidase, an enzyme which is only functional in senescence. So in this way, you can have this. It has been observed that we have taken animals. When you take animals at different age profiles, you study the cells, you find a progressive increase in these cells. Once again, highlighting the fact that if at least not a causation, there is a causality, definitively. So this is how they look like. And now coming to what we have done through dietary factors, I used green tea EGCG. We're all familiar with green tea. It's one of the favorite drinks to have. And it is rich in something called anti-gallate. So now what I did here was took this and gave it to weaning mice lifelong. For about 20 months, day in, day out, this experiment ran. And every day I gave the animals EDC. And what we looked was how it impacted with age. So you can see 6, 10, 14, 18 months. We profiled to figure it out. I just want to show sir this here. You see this here is the DNA damage response. So you can see here in 6 and 10 months, there is nothing such like that. There is no damage in the animals initially. 10 months of a mice would probably be like 40, maybe 50 years as far as translation to humans is concerned. And then there is a sudden increase in the damaged DNA response. Now imagine these animals lived in captivity, never saw any real world threat, just aged naturally. When they still, there is a lot of damage. And those guys which were given EGCG, they had lesser damage. So anybody, even in a non-scientific terms, having good DNA, good genome translates into a healthy cell, which translates into a healthy tissue and an organ and then an organism. So we protected from the DNA damage and then some markers of senescence like this here is the cellular senescence marker. This indicates that the cell is no longer dividing. So what happens when a cell is countered with damage? When any of our cells get countered with damage, it has 2, 3 options. One, it fights with it and it overpowers it, it's healthy. Number 2, the damage is so much that the cell will be told, okay, you undergo apoptosis, kill yourself because you're damaged now. And then there is a fine line. The damage is just enough that the cell doesn't die and it enters the state of senescence. So you want to protect the cell from damage. And you can do that with the help of something like polyphenol, which have the ability to fight the stress. So this here, in this way, we protected, we observed in different tissues. I'm just giving you representation of adipose and intestine. And some of the effects in the plasma, these are cytokines related to inflammation. See, when do we get inflammation? We all suffered through or went through COVID. Inflammation was only high, right? So these animals, we just ate. Look at the levels of inflammation. There is nothing here. And it started to increase without any apparent cause. Why should that be? Because the body itself is feeling inflamed. Without any cause, these animals were just raised in captivity. And as they aged, they had higher levels of stressing cytokines present in the plasmas. And once given all those who went through the polyphenol EGCG, the levels decreased significantly. So once again, it tells you the relevance of studying the senescent cells, and number 2, how phenols can have real world effects, right? So, okay, this I wanted to, again, we are currently now focusing on medicinal plant, Picrorhiza curua. It's a plant found in the high Himalayas. And people have been using this for generation to counter diabetes. So we wanted to study how it works, what its role is. So here, just a simple observation. Again, this is DNA damage response. You can see the senescent cell here, if I could perhaps zoom in. These green, this here represent activation of DNA damage, and it is pretty much there in all the aged cells. All aged cells have DNA damage, invariably. And they still do not die. They remain there. So that tells you that if they persist for long enough, they're going to cause trouble. So you can see over the insert here, this indicates DNA damage, and you can see progressive decrease when the medicinal plant was being used. So this one way, then another way is related to proliferation. All aged cells do not divide. So more the green, the more dividing they are. So you can see that when treated with the plants, their division here at least is very similar to it. So again, the effects are being nullified again through something which can have, which is dietary element, the nutritional element. So apart from that, we are also working on metabolic disorders. This here is our cellular system we have developed indicating adiposity, so adipose tissue obesity. And again, using the plant, we have developed a new kind of formulation, and it is effective in that. We are currently into trials for this. And we also are working on sarcopenia. So again, we are using cellular models. So this cellular model, this cell, you can see here, there are lots of nuclei here because the number of cells are fused together to make a fiber. See, this is the fiber being formed. So we have developed a system to study sarcopenia because very much associated with elderlies. So how sarcopenia can be countered using the dietary elements. So again, something we're still working on this. The second element apart from polyphenols we have is probiotics. Now this becomes very interesting. When you talk about bacteria or probiotics, they survive in your gut. Now see, our immune system is designed to find any bacteria and just destroy it. But these bacteria survive in our gut. They are in constant communication with our physiology. They release certain molecules. They never cross the gut, but they release molecules which are called postbiotics, which can enter circulation. So we want to study whether because these probiotics, we are meta-organisms, we have more bacteria in our gut than the entire cells of our body. So bacteria actually own our bodies more than we do. So the metabolites that they produce, they have their own effects. Now they are already there in your body. So what are their effects? So we wanted to study their effects on aging. And this was the first report that we hypothesized that they might have some effect. And they actually did protect the adipose tissue cells from senescence. Again, these works are published. So these indicated that if you have good bacteria, the so-called good bacteria, the probiotics, they will help protect you from accelerated or premature aging. So this is what it indicates, at least at the preclinical level. So again, this stems from the fact that the gut microbial, the metabolites can cross through, and they can cross through very effectively if the epithelium is damaged. If the gut epithelium is damaged, the movement is even more progressive. So in aging, this happens a lot. So more and more metabolites can communicate, and it can be reciprocal. These guys can also affect them. Very intricate relationship that we are trying to decipher also. So how do these two interact? These two phenomena interact. So the gut metabolome, the entire metabolomic pathways and the molecules produced by the gut, they can have their physiological effects, including age. We also developed fermented milk, so to speak, and we tested its efficacy against immune aging, another important component that we have been doing. The intention here was to again find out whether specific probiotics, the intention is to how to identify that probiotic that I'll discuss in a moment. So how to identify, and once you have identified, study their effects. These are again the effects related to natural aging. As we are aging, we have increased levels of inflammatory molecules which are being suppressed through the treatment with the probiotics. So individually, we have been working. The key thing which relates to innovation. The first part here described what are the basic components that you can target. Suppose in this case, we are using aging as a kind of a vector, kind of a disease. And then individually elements we discussed, probiotics and polyphenols. The key thing which leads to translation is that you can combine them together to create what is called a symbiotic. It's relatively new, but very upcoming market because you can very meticulously combine these two together. So I'll discuss in a moment here. When you probiotics are good bacteria which you require, and certain things help in their growth. Those are referred to as prebiotics. So when you have combined them together, the product becomes a symbiotic. So if somebody says, I have a symbiotic product available, it will have the bacteria and the component which accelerates the growth of that bacteria so that this bacteria survives better. So the idea is to create that kind of product. So the current symbiotics will be defined as mixture of live microorganism and a substrate, which is selectively utilized by the host. The keyword here is selectively. We'll discuss what it is. And the component one, the first component, of course, will be the live microbe, the lactic acid bacteria, and the second component, which is the this substrate. Traditionally, this substrate is this. As you can see, these are inulin, oligos, and dactylose. These are some kind of sugars. So you give sugars, the bacteria grows, proliferates better, and it survives better in the guts. That was the intention. That is how you create this symbiotic. These are actually further into two classes. I'm not going more into technicalities. A complementary symbiotic will be when you just have these two combined together. A synergistic would be that the second prebiotic component is selectively increasing the growth of the probiotic. You have specifically selected it to support the growth. Here, these two may not be mutually very synergistic, but by definition they qualify. Here you have actually identified that it specifically increases the growth. This is called a synergistic symbiotic. This dichotomy is still being developed, and the International Association of Probiotics and Prebiotics is trying to delineate it. So how these two can be better defined? It was just very recently they created this dichotomy because there was a lot of confusion among the scientists as well as the consumers alike. So what actually is a complementary or a synergistic symbiotic? So when you combine any two of those together with this properties, that's a complementary. But if these two specifically are being shown to affect each other, it's called a synergistic. So with this, now we move on to what are the challenges. Currently, the challenges we have non-specific microbial. Since the sugars that we are using, they increase the growth of probiotic, but if the pathogen gets an opportunity, it's also going to grow in it, right? So how do you justify that? If somebody has lots of pathogen, their gut is imbalanced, and you provide a prebiotic, it might help the growth of the the pathogenic substances. You certainly don't want that. Some inconsistencies were observed in clinical studies. Inherent, that's very important. Lack of inherent what activity or bioactivity does the sugar have? Only towards nutrition, there's no pharmacological effect of its own. So when you're giving a symbiotic, which has two components, one of them is pretty effective. The second one is not. Second one is only helping the customer. What could be a better symbiotic in which both have inherent bioactivities, created more pharmacologically beneficial? So this here relates to that we need new sources of prebiotics, new sources which can help in the growth of probiotic, but the product that is being formed is actually been more beneficial than the traditional ones. So that led to this, that polyphenols can be used as symbiotics, and this is where the game changes. So currently, if any symbiotic is available anywhere, you will see it will have sugars in the prebiotic. And what about polyphenols? Polyphenols are usually considered or traditionally considered as suppressive, microbial, antimicrobial. People would take them as antimicrobial. But if you dig a little deep, their effects on gram-positive and gram-negative bacteria. So what they do is they suppress pathogens, but may exert growth-promoting effect. Even if they don't exert growth-promoting effect, they will definitely suppress pathogens. So what it will do is it will create a niche area. The gut is a very prime space for the bacteria. Everybody wants to colonize everybody's guts as far as the bacteria are concerned. So when they say, okay, some pathogens have been cleared, the space is opened up, let's occupy that, right? So in that sense, you can have good bacteria, and the gut balance changes or shifts, right? So we are utilizing the properties. So of course, we need to find out which polyphenols can do that. It doesn't fit everybody. This is where we come in. This is where the technicalities will come in, as I should discuss further. So important. So polyphenols can suppress the pathogens that allows the growth of probiotics. So this in this way it qualifies as a prebiotic. But this is not where the game ends. The bacteria can then metabolize the polyphenols. If you remember, I initially discussed that polyphenols, howsoever good they may be, when you consume them, they are very large molecules, very difficult for the intestine to absorb it. So whenever you eat anything, the first one who is feasting on it is the gut bacteria. You might feel it's very tasty and everything, but trust me, inside the bacteria are having a blast of it, because they are the ones who are going to actually consume it first for nutrition. And in that process, they're going to break the molecule, make it simpler so that you guys can absorb it. So the polyphenols, which are very potent, cannot be used by the body because they're large molecules. Probiotic is going to break it apart. So you can see there is a reciprocity here. If you can identify a combination in which the polyphenols increases the growth of probiotic bacteria and the probiotic bacteria suppress, metabolizes the polyphenols, that would be a win-win combination. This is what we are working upon. This is what we have identified, and this is what I'm trying to sell as innovative functional foods. So there is this reciprocity, and as you can see here, polyphenols might suppress pathogens but augment probiotic bacteria. The probiotics in return break the polyphenols and they make a better medicinal plant extract, if you will, a better polyphenols which can be used as foods. So with this idea, we are actually pioneers in this and trying to get the community, consider this as the opportunities where you can create new and innovative functional foods. So I just two case studies I'll discuss. One I have already completed, and second one is in continuation. I'll discuss few bits of it. So, for example, I studied a complementary symbiotic. As the name suggested, you just combine two ones together, any prebiotic and any of those. But I was not interested in sugars, so I took my favorite EGCG, the catechin that we discussed earlier. I took this, and first I studied what is its effect on bacteria. All these guys shown in red are the pathogens. So you can see in the presence of EGCG, you can see the growth minus. Everything is decreasing. But look at the bacteria, not so much, probiotic bacteria. So the average inhibition of pathogens is above 50%, and this is just 14%. So if this is a limited space, what will happen? These guys are stressed out, so these guys will take over, right? This is one way of doing things. So once you identify, the key thing is identifying the right combination. The key word is how do you identify which bacteria and which phenol to use? That is a key takeaway. So once I identified this, then I tested here. Sounds pretty complicated, but the simple thing is you gave the cells some stress, and this is how the EGCG protected. This is the stressor, and this is the catechin. But you see, when I combine this with one bacteria, the effect is a little enhanced, significantly enhanced, maybe like 25% or so, but still. So it is increasing the effect. So I thought, okay, this bacteria is my choice here. So you can see the intention is to identify which polyphenol and which bacteria. So once you had that, then you can do your further studies. I did this to aged animals, as aging was my forte here, to EGCG and the bacteria. So you can see here, this is what control is. This is when only the T catechin is given. This is only when the probiotic is given, right? And this is when they are combined. See the effect? Can you see the effects? So these are clinical kind of preclinical effects done in animals. The effects, additive or synergistic, when you combine them together, it becomes complementary. Look at this, NRF, the antioxidants. The antioxidant response is pretty much higher here. It is strengthening your antioxidant defenses, which will have an impact on how you counter or disease this. But the inflammation is going down. So this work paved the way for us to file for this. We got a US and UK patent on this because green tea is consumed worldwide. So I was saying that we do not just consume green tea, combine it with the lactobacillus, the probiotic that we had, should have much better effect. So building upon these ideas, we are now working on a synergistic symbiotic to take the game further, to study further. Here we observed that here we are defining the prebiotic activity score. We took different types of medicinal plants, found all in high Himalayas. And we're trying to assess whether they can act as a prebiotic. That means whether they can support the growth of probiotic bacteria. And out of many of those, only two become the potential candidates. And these two plants, this again, this pictorizer that I just mentioned, and this Teratoda vesica is a plant extensively used for COPD, asthma, allergy, respiratory disorder. This also looks like it has an effect. We studied their effect on using animal fecal matter we used, and we used to identify lactobacillus. And you can see in the presence of the plant, the growth of the lactobacillus where it is going. And these plants suppress the growth of a pathogen E. coli. So it's a kind of model pathogen that we took to identify. This is still ongoing. And the another thing that I mentioned, remember that there is reciprocity. That means the probiotics can suppress, probiotics can induce changes in the profile of the plant. This is LCMS of the native plant. And here what happens when we add probiotics into it? There is an increase in small molecular weight. We are just to characterize all this, because work is still ongoing. Increase in small metabolites. So degradation of the large metabolites into smaller ones, which resulted in this is called biotransformation. So what I was trying to say is that probably we are getting the bacteria is breaking apart the plant, utilizing the plant's resources for its own survival. Now think about it. A plant, something like this pictorizer, is a highly iridoid molecules rich plant. It has lots and lots of high polyphenols. Nothing survives in those polyphenols. When we did this, the experiment lasted only for 6 hours. Bacteria could not survive. But in those 6 hours, they utilized the plant, did whatever they wanted to do, increase their abundance, increase the polyphenol content of the native plant, made it much better. The plant, which is already so potent, has now been made even more potent. If you want a number, perhaps like 25% increase in its activities. I'm not shown here those activities, they are still being done.
The intention is if you have the right combination, you can create a right product and increase the value of both the native plant as well as the probiotic area because it grows better in it. So this results in creation of a second generation. This is called a second generation of prebiotic and a second generation of synbiotic. No products like those are available. You will never not find that you have a fortified plant added or a fermented plant or a combination. So this is a very open area. As far as I researched a little bit, I find that you guys also have lots of medicine plants. So already they are shown to be effective. So can we create a combination and make them more effective? Can we look for fighting against, say, anxiety? Can we look for metabolic disorder? And this will, I think, I certainly believe in it that this is a game changer. Today, tomorrow, this will pick up. Symbiotics are coming. If you tell the aware consumer that you have a product that has medicine plants as well as probiotics, it helps your blood. It increases the potency of the medicine. There is no shame. There is no reason to believe that this might not be effective to the person. So the pipeline could be something like this, as I did. This is how you can do that. You can have prebiotic. You can actually have your own probiotic. Yeah, that's important. Where do you get the probiotic material from? As I already mentioned, they have different. They're different. So probiotics in India might not be good enough for this kind of population because your microbiome is different. So why not isolate from you guys or whatever you are consuming as probiotic foods? If you take these probiotics and you combine with these plants which are found in your area, it's a win-win combination for the plant as well, for the nature as well, and for the industries as well. And yes, I'm working on this as well. Why this leaked to me was because the plant that I'm working on is highly bitter. It's very bitter. And when it got fermented, it was even more bitter. I was like, you could take it. Why do you make tablets or sachets or pouch? That's kind of winning as well. And perhaps not that appealing, not appealing. So I said, okay, let's take a delivery system. We already have a product, perhaps dairy can be used. And New Zealand is a very thriving dairy sector. So maybe we can take the product, combine it with the milk, and research has shown that milk is a carrier of polyphenols. It can carry it as well. So you imagine having a fortified milk, which is 45 million medicinal plants, probiotic bacteria, and is targeted for satiation or any other thing that you might have. So this is really something, and it aligns well with dairy as well. So this is kind of the thing. So it has a lot of market potential, prebiotics, probiotics, symbiotics, all are growing, especially symbiotics. Even now, I think there are resistant symbiotics. I'm not sure if there are any symbiotics available product that might be there. We are talking about synergistic symbiotics, next level of symbiotics. This really adds novelty to it, and it's growing rapidly, and rising consumer awareness is even likely to push it further. So with that, ladies and gentlemen, I think what I'm trying to say is this. This is what I'm trying to say. You guys are better together. The only thing is we have to, they need to gain a little bit more. So they should not like each other better. So you need to find out which one likes the other one. Once you are through with this, you can probably create new functional foods which can be fortified with these plants and then can be used against certain disorders, maybe like aging or anything that you might aim at. This is how the amalgamation will work. As the title of the study says, the amalgamation. The amalgamation is not random. It has to be scientifically identified and proven. We have two of those. This is a developing field. It's very interesting as far as innovation in functional foods is concerned. Create these, all the fundamental mechanisms are still to be elucidated. How do these two interact? What makes one bacteria interact better with the other one? What genetic, epigenetic, or metabolic changes? Many times people can have an interest, and partial people obviously will have an interest. So, having said that, we look for any collaboration, anybody can be interested with the faculty, or any exchange, faculty, or something like that. Any funding, international funding opportunities, considering the scope of it in New Zealand or anything else that might fit to that. I would like to acknowledge here at Lincoln University, the department, he was communicating with us regarding this travel grant that I was given through Dr. Gagan Jain. So I'm very grateful. I'm very grateful to the University of Lincoln, beautiful place, beautiful people. Really enjoyed it, although still jet lagged, but I am kind of enjoying it. So I would also like to, at the moment, take a moment to acknowledge my funding partner. These guys have been giving us funds to do whatever we have been doing. Apart from that, we come from this place, this Solan, which is a picturesque university that we have high in the Himalayas. And we have lots of Himalayan diversity, biodiversity available with us, and we try to tap on it. So we are developing functional foods. We are developing phytopharmaceuticals. This is our small but young lab. We are trying to do as much as we can with the resources we have. We are also working on basic themes like I already mentioned. Now, actually, we have a hospital attached to do clinical work as well. We are doing that. We have facilities available to take care of animal cell culture. We have a food department, food department, have packaging, have create the product in the labs to showcase it to people. Maybe after a year or so, if I come back again, I would have that one of the products that I'm developing. So maybe that is something. We do a lot of cell biology, molecular biology. So we have all this stuff available to carry out. Whatever research I've shown you, I've done in my own lab. Any questions?
Sharma, thank you so much. Any questions?
I'll start with social acceptance. So when you said you're choosing that garden medicinal plants, and local people are already consuming those medicinal plants, so your research is enforcing the usage of this medicinal plant. So how the local people are accepting? What's the social acceptance?
Yeah, local people feel like that actually somebody is doing something. Because it will also have an impact on agriculture. Somebody, but some industry or two tomorrow use that plant, they will need more of that plant, right? So we tell people that, okay, give us that stuff. If this comes out to be good enough, there will be more agriculture on that also. And people are like, okay, you are developing something out of this because we know it's effects are there. Nobody, later down believes us. So if you're doing something, we're okay with it. And now we are telling them that whatever plant you're growing, I'm making it better. I've already made it 25% more potent. Now the results I couldn't show you because they're still going on. But I think in the 6 months or so, we'll have that. So I think it's a combination. It's really because it's natural. Both are natural ingredients, bacteria as well as the more than anything, we just finding the right combination and hoping that the marriage doesn't end in a divorce. You can win.
So are you involving the local people?
Yeah, we are. We are involving the local people. We tell them, they tell us, because they know many secrets that they don't want to share. They feel like that, okay, that these guys are really doing something useful. Sometimes they give us some of the tips which really go along. For example, somebody said that, okay, don't take the plant from there, go to that side. Those are more preserved. So we actually took and we saw what is the difference. There was a difference in their potency. Right? So the plant can be found in lower hills also. Like, please don't do that. Go to the harsher hills. So my team went to the harsher hills, telling everybody that we might also be lost sometime. Took the person with us and then we collected some of the plants. So I mean, this is how they were.
I think we are very similar in collecting data that the local people.
Yeah, those guys know better. Yes, those guys are. Yes, sir.
Thanks for coming to Lincoln. Why did you choose EGCG? Because what we find is, the polyphenols like resveratrol and just more effective. Effective in the sense of effective, we don't forget, but the growth wise, they appears to be better. Both of probiotics.
The resveratrol. One of the reasons was I worked at I worked at the Himalayan Institute, which grows tea, Palanpur. It's a place where there's a lot of tea production. If tea comes to mind in India, we think about, I think, Darjeeling, perhaps some of you might have heard. But there's Kangra tea also there. Nobody worked on that. And then the second is, it is rich in EGC. About 60% of it. So we wanted to study that. Resveratrol, I mean, might be yes, but we found that EGCG is also growth promoting. And as you said, it is already known. To make any point, it's already there. You tested a new phenol. That's what I'm saying. We should test more and more about. These are not being tested. People study them as antimicrobials. But as you dig these, you realize they interact in many different ways. Here I have just shown how bacteria interacts with the phenols and does to that. Phenols can also have an effect on bacterial metabolism. And it is also an active area of research. We try to find out how phenols are affecting the fighting. Please keep in mind that whatever we do, regardless of we do something about it or not, consume phenols to the bacteria in the gut, they will always interact. It's not something that, okay, I'm a vegan, whatever you are, all have bacteria. And one form or the other, we are all consuming phenols. There will always be an interaction. Not specific, it's not very typical, let's say. Right, sir.
It's a really interesting talk, particularly prescribing for in vivo studies, the live animals.
Yeah.
So many of the studies are right. For the GCG, you see that sugar was one of the larger polyphenols expected to go down. Do you have any idea what it's taking up? What was absorbed?
Okay,
Which part?
okay, so it gets extensively biotransformed in the gut. Glycosylation, decarboxylation. These are the things that gets broken apart. When we try to measure it, actually I didn't do that. When we try to measure the levels of EDC in the plasma, right, we end up getting the glycosylated form, the broken apart form. Right? For that, we need to find the kinetics of it. Right? Is that what you're asking? We did it.
Yeah, it's a still contain EGCG with extra sugars rather
Yeah.
than broken up.
When they are broken apart, they may be glycosylated. It is broken apart, and probiotic bacteria, they have enzymes to break it. And they convert it into smaller forms. Usually what we do is if it is big one, we look for a chemical reaction to create what kind of product. So if the plant has a glycosylase, break it apart into two smaller molecules. You do the LCMS and try to identify which one it would be. So simple diamond, acid, yes. Thanks.
Thank you for a really interesting insight. I was wondering, how those polyphenols would suppress the pathogen.
Yeah, that has to do with the membranes, how they interact with the GCG. When we observe this, we also ask the fundamental question: how do bacterial species acting differently? Initially, we could only, there is no specific study of this, but we found out that there's something between them, gram positive or gram negative. This is what the difference we could associate them. Gram negative bacteria are more sensitive to these as compared to the others. This is what I think their absorption is different because then again, this is another line of work. That how they interact. For example, somebody wants to study how bacteria interacts with plant and how they interact. So this is their. We were more concerned in initially finding out which one is the association which benefits us. We didn't go into the deep into that, but that's again, as I already mentioned, fundamentals are still to be asked. Why these two are interacting in a better? Why not the other one? Right. So this is for fundamental people to further decide, maybe microbiology. There is more on pharmacological side. So I just said, okay, these applies to that. So, and then next. Yes, sir. Yes.
I have a few questions that's why we did it. To understand the context, I want to understand, I want to know first that the cell line that, can we go to the slide where you showed negative form? Yes. No, not this one. This. So to understand the context, the cell line that we use, it was an embryonic stem cell.
No, this is not a cell line.
Not here, like this beginning that the one that you showed, 3 piece.
Yeah. That is, it's not an embryonic stem cell. This is taken from an embryo. This is not a stem cell line. PT3 album is here. I think the first one you have this one. And this is taken from an embryo. embryonic sensor. This is taken from a proliferating embryo. And that is why we could use it now, because the embryonic sensor will proliferate pretty fast. So because otherwise is a terminally different. You can't do that.
So how many generations does it take to end its cell division?
Naturally, we are asking replicated cells. We might have to go about 70 years.
70 generations, and what you found out is in 7 generations, maybe like in 7 days, it dies if you go through H2.
Okay, that is no. For that, we are not using replicated cells. Good that you asked. We are using a stressor. This is called a stress-induced premature senescence. If you were to wait the cell to divide, it will keep on doing that for 70 generations, right? It's going to take probably a year for you. Please remember, it's an embryonic cell. It divides pretty fast. It's very similar to a tumor cell. The only thing is it will eventually stop, but not a tumor cell. So what we do is we don't have to wait for all that long. Remember, we are trying to do a HDS here. I have 10 different plants. Otherwise, I have to wait for a year. So I said, okay, this is obviously naturally you would want that, but we are all in stress. Be it any kind of stress. So I thought, let's give a metabolic stress. Once you give the metabolic stress, the key here is this. See, you can have an acute response, which will look similar to aging, but it is not aging. So once, see, the stress is being given for 3 days, and then we keep the cells 4, 5, 6, 3 more days, and we study on the 7th day. If there was any superficial response, it might be gone here. If there was any acute response, it is gone. But at the 7th day when we studied, and we tried to replicate others' protocol, it didn't work. People usually would give the stress for 1 day. It didn't work for us because it was not stable. So we said, okay, aging doesn't happen overnight. Nobody sleeps young and gets up with its years old. So let us give it again and again. We developed this model for 3 days, and we gave enough stress. Even if you increase it more than 3 days, the response becomes more and more stronger. But the thing was, you want the response to be stable. We have observed, we have kept these cells for at least 30 days. They just stay there. For those who are familiar with cell culture work, if you keep the cells in media, they will keep on dividing. They will proliferate. They will contaminate everything. These guys just stay put. All you have to do is change the media so that you give them, they just stay put. Nothing happens to them. They show all their features, which shows that they are characteristically cells. So we have developed this protocol not only for this cell line, 3D, we have developed for liver, we have developed for muscles, and we have developed for lungs as well. It works. Even I didn't show it, I didn't want to make it too scientific. It worked on terminally differentiated cells, macrophages. Because I'm also interested in how macrophages undergo cells. It's a very interesting topic. Immunological aging is an entirely different beast. We are trying to take it as well before we need to understand what it is.
So my question about this is like the standard experiment that you developed on which then you started trying your
Anti-aging or anti-senescence. Yes.
Can we go to the slide where there was negative?
I almost feel like why you shouldn't have transitions. It takes time to read. So here we are.
Can you explain how this was?
Okay. So basically, what we did here was you took these are all bacterial species, right? Like if I remember correctly, Vibrio pulveris, Staph aureus, Klebsiella, and so on. These are all pathogens. You grow them in their normal media, and you add PDCP. 1%, we made a profile 0.11%, so on, and see what happens after 24 hours. You measure through CFU, colony counting, as well as absorbance. You can do that, and then we find out what is happening. So we found that these guys were strongly suppressive, inhibited like anything. So this was this revelation for us. Because until then we had only read DI, so it was like that, but we never really got. So
the way this graph was created is based on a control where
it
was not
it is.
then over here it showed negative.
Yes. It's percentage, right?
One last question.
Yes.
So as we discussed, like probiotics interact with the polyphenols and converts those polyphenols into some metabolites like probiotics, so that might be the probiotics producing certain enzymes. Yeah. So what if we use just the enzyme cocktails? Use those symbiotic.
Yeah, you can certainly do that, but it is not going to be costly. You have to isolate the enzyme, that is one thing. But certainly you may have one advantage, because if you put out the availability of the probiotics, right? You have isolated any number. That's all good, right? But just to start, because it will not be symbiotic anymore. Definition says we must provide live bacteria. If you were to take a component out of it, then you cannot claim that the product is symbiotic. Of course, it's only technicality, because it's likely that you will get the end result. But again, if you claim the bacteria, you may have a consortium of enzymes already available, right? One enzyme is good for all. So because the medicinal plant they have many different types of chemicals, right? And it might buy more than one enzyme, right? So scientifically, technically, you want to do that out of it's okay. I think from a translational perspective, it will be perhaps more useful if we can go with the guide.
Dr. Sharma, thank you so much. Thank you very much. It was a pleasure that in Shoolini's event of the Center of Excellence for Transformative Business. I think we have another event on Friday, and I think next week we have some more events as well. So do go to our website, transformative.business.co.nz, to look at all these events and judge. Dr. Sharma, thank you so much. I know you will be in the country and I think university for the next week and a half. If there are any more questions in the room, are they allowed to contact you?
Yeah, please. That's what any questions. Anyway, I can.
So feel free to contact Dr. Sharma.